Hi, my name is Emel Küçük, and in this poster I present research on the neural effects of TMS over the prefrontal cortex in emotion recognition. Emotion recognition involves a network of brain regions. These include the superior temporal sulcus, which is involved in processing dynamic aspects of the face, such as emotional expressions, the fusiform face area, which is involved in recognizing faces and identities, the amygdala, which assigns emotional significance and aids in detecting and responding to emotional stimuli, and the anterior cingulate cortex, which is involved in the emotional regulation and cognitive control. And of course, the prefrontal cortex, which modulates activity in the amygdala and other limbic structures. Now, impairment in these regions can result in impaired emotion recognition, and a disruption within this network has been implicated in various conditions such as anxiety and depression and autism and schizophrenia. The aim of this study was to investigate how disrupting the prefrontal cortex impacts neural activity in other brain regions when recognizing fearful and happy expressions. 20 participants performed a delayed match-to-sample emotion recognition task in the MRI scanner after TMS was delivered to the vertex, which was a control site, in one session, and the right inferior frontal gyrus, which is part of the prefrontal cortex, in another session. Participants were presented with the word happy or fearful, followed by a face depicting a happy or fearful facial expression. Participants indicated whether the facial expression matched the previously presented word. Disrupting the prefrontal cortex compared to the vertex had an effect of increasing activity in the right inferior frontal gyrus, so disrupting the right IFG could paradoxically lead to increased activity in this region due to an attempt to compensate for the disruption. Similarly, right IFG disruption may elicit compensatory mechanisms in the right SDS, leading to increased activity in this region as well. There was a relatively stable activity in the right FFA, which is likely due to the fact that the task was an emotion recognition task, not a facial identity recognition task. There was also an increase in activity in the right amygdala. Disrupting the IFG may decrease its regulatory control over the amygdala, leading to heightened amygdala activity. And there was also a decrease in activity in the anterior cingulate cortex. Decrease in activity here after IFG disruption corroborates findings in studies investigating depression and anxiety that demonstrate functional connectivity between the prefrontal cortex and the anterior cingulate cortex. It's important to note here that there were no behavioral effects and no effects that were specific to happiness or fear in this study. So the key finding from this study is that disrupting the prefrontal cortex increased activity in the right amygdala and decreased activity in the right anterior cingulate cortex. The findings highlight the role of the prefrontal cortex in modulating activity in regions associated with emotional processing. The results have potential implications for understanding and treating emotion processing disorders, as the findings support the use of the IFG as a target for neuromodulatory interventions. Increasing the difficulty of the task may also provide a greater insight into the behavioral effects of disrupting the prefrontal cortex during emotion recognition. As in this study, the task was very simple, and this simple task meant the demand on the prefrontal cortex was minimal in this study.